Hi, Ben.
Parrish, how are you?
Very good. Thank you. Thanks for having me.
Yeah, thanks for being here. It's great to see you again. So, please let me set the stage for this. Your case study centers on changes in methods and production processes to get a product to market, which otherwise may not have had that opportunity. Before we jump into that little teaser, if you wouldn't mind, give us a little bit of bio and background on yourself.
So I've been in biomanufacturing for over 40 years. Started with 4 different drug companies for the first 20 years of my career. And then in the last 20 years, I developed or started a single-use technology and services company called Accelerex, and then sold that company to GE a couple of years, or sorry, 7 years ago. And I'm an independent consultant now in the bioprocess industry.
Got it. Thank you for that. So the title of your case study is Getting to Yes, How Major Changes at Phase 3 Can Make an Unapprovable Product Approvable. So if you wouldn't mind, maybe paint a little bit of color into that picture. What exactly is all this about for the audience?
So this is about a drug that had made its way through clinical trials and was in fact in the middle of phase 3 clinical trials. And it was showing great promise against a fatal neurological disease. So the clinical folks at the FDA were very interested in the product. And as a result, they began looking at the commercializability or the licensability of the manufacturing process and found that it was severely hampered or inefficient and actually making unsafe drug. So they came to our company and asked us to think about how we would completely transform the manufacturing process to turn it into a licensable process to produce the drug. So that was the challenge that we faced.
So now that's interesting. It sounds like licensure wouldn't have happened if the changes weren't made. And maybe as you go into a little bit deeper of the explanation, consider sharing for us any information you could about what sort of delay might have been incurred if the changes weren't made. Could it have been a delay of many, many months? Maybe licensure wouldn't happen at all. And, we also have to remember there's a time value of money. There's a cash burn rate associated. So, the longer products are waiting to get to market, this is costing tremendous sums of money for the sponsor company. So maybe keep that as part of your narrative as you explain a little bit more of this story. And so maybe, I guess, jump back in on what was the nature of the production process and what sort of particular leeways did it afford you as far as what the FDA was looking for what a well-characterized process should be? How were you able to make changes?
Right. So the problems with the original process were, number one, it used a human foreskin fibroblaст cell line. So there was a great variability in the productivity of those cultures as they came from the hospitals where those foreskins were being collected. So that led to a very variable production output in the manufacturing process. The second limitation was that the cells were grown in attachment-dependent cell factories,
right,
which don't scale up very well. In fact, they have a limited scale. So there was a scalability problem with the process in order to meet market demand. The third problem with the process was that it had a lot of serum in the process, and that confounded purification and resulted in a low purification yield. So between the variable in production amount, the serum, and the downstream purification low yields, the cost of manufacture was very high, and the output was very variable. So those three things. The other aspect was that the cell line actually led to a safety risk in terms of human virus, in the final product. So those four issues were really standing in the way of this process being licensable. Could it eventually have been licensed? Well, we'll never know. But the point was that the FDA was committed to getting it to a licensable state and was willing to take the risk that the new product made by the new process would not be the same as the old product. So that led to a whole series of discussions and challenges as to how to ensure that the new product was in fact safe and efficacious.
Right. Right. So in order to get through that hurdle with the agency, then that brings us, I guess, internally to your three goals. So the goals in order to get to that point were to increase the process robustness, reduce the manufacturing costs, and validate the commercially viable, scalable process. So how did you go about accomplishing those goals? First of all, what was on your mind in targeting those three goals? And then what was the next step?
Well, the first was to get rid of the problem with the cell line. So we decided to clone the gene for the drug into CHO cells and to develop a suspension process and to develop enough knowledge around that process to understand what would affect variability in manufacturing production amounts. So we put together a process development team to do that work. The second challenge was to get rid of the serum in the process. So that required a lot of development work to wean the cell line off of serum. Thirdly, by getting rid of the cell line and changing it to CHO cells, we were eliminating the human virus risk. So that was another big advantage of doing that work. And lastly, overall, we improved the purification yield. By getting rid of the serum, the purification yield increased substantially. So that helped on the manufacturing cost of goods side. So those four objectives were the goals of the process development effort to develop a new commercial process.
So your selection then was the development of the CHO suspension cell line. It was a 2,000 liter scale stirred tank process.
Right.
And then modifications to purify serum-free product.
Correct. In fact, we built a whole new commercial scale facility to produce the drug. And in that facility, we produced material to enter into a clinical evaluation that we negotiated with the agency.
Excellent. So then in all of these, I mean, really deep and complex endeavors that you were engaged in, you now get to the point where we're at the main thrust of this case study. So taking an unapprovable product and getting to this yes decision, adjudication from the agency. So what were the results at the end of the day?
Well, the results were that we reduced the manufacturing cost by about an order of magnitude, which was very substantial. Number two, we improved the scalability by developing the suspension cell line and the suspension process. Number three, we improved the purification yield by getting rid of the serum. And the purification yield improvements helped us increase or reduce the cost of goods substantially. So the real next question was, well, that's all fine and good, but what about the quality of the product? Yes, it was safer because it did not use a human cell line any longer. But what about the activity and safety of the drug itself? So that became the next big question to be addressed.
Hmm. Interesting. Now, as far as the results were concerned, um, did you find that it was an easy sell when you had the results in hand to be able to get to the goalposts that the FDA was looking for? Or did you find that there were also other obstacles along the way that you hadn't considered?
Well, the one obstacle that showed up was that the molecule had substantially different glycosylation pattern compared to the original molecule. So we spent a lot of time in process development to try to um, modify the cell culture to change the glycosylation to more closely match that of the original drug. So there was a lot of angst about that because glycosylation can affect the half life of the drug. It can also affect the potency and efficacy. And so these were viewed as very substantial risks in developing this new process. And we spent 50 man years, literally 50 man years of process development time to try to modify the process to get it to produce drug that was similar, more similar, or more comparable to the original product. So that was a lot of effort and a lot of concern and a lot of risk that had to be faced in doing this project.
Hmm. And then, as far as what the agency requires or requests from a well-characterized process, is there anything within the regs or guidance that um, was that allowed for you to be able to make these changes more easily, or on the flip side, that made it more difficult for you?
Well, we negotiated an approval process, which would include first doing a biochemical comparability between the new product and the old product. And and if the answer to that was that it was not comparable, then the next step would be to go to sort of phase two, which would be to test the drug in animals and look at the PK/PD performance or behavior of the new molecule versus the old molecule. And so
Right.
we had to produce material to do that study. And the study showed that there was comparable PK/PD between the old molecule, between the new molecule and the old molecule, despite the fact that there were significant differences in the glycosylation patterns of the molecule. So that was a huge relief.
Hmm.
If that had failed, the next step would have been to do a more involved human clinical trial. Um, so we were able to, as a result, negotiate with the FDA to conduct a limited bioequivalency study in patients to study the PK/PD in human patients and to also look at efficacy. So that's in fact what was done. The trial was conducted with 17 patients, and the efficacy was evaluated for the new molecule. And
Hmm.
fortunately, it came out comparable to the old molecule. So that was a happy ending to the
Yeah.
old project.
A very happy ending. Any parting thoughts you have for us about this case study that you'd like to share? And remember, to the audience too, for those who may find themselves in a similar situation with changing of a process or changing different methods, just remember to head to the networking session at the end of the panel discussion. And please find Parrish at the appropriate table. Parting thoughts on this, Parrish?
Well, I think that changing the process so radically in the middle of phase 3 back then was a watershed event. Nobody had really done that before. Changing the cell line certainly was a verboten in the business of biotech at the time. So, this required a huge investment by the company to commit the resources to do the development work. As I said, 50 man years was spent doing the project. So, if you're anticipating changing your process in phase 3, sit down with the agency and negotiate a series of comparability steps from level 1 to level 2 to level 3 to be passed as you develop the new process. Comparability level 1 being the biochemical characterization of the drug compared to the original drug. Comparability level 2 being behavior of the drug in animals. And comparability level 3 being comparing comparability in humans. So today, those comparability protocols are pretty well accepted. And as a result, it's led the way to a more defined path for companies that want to change their processes fairly radically on the way to the market.
Beautiful. I couldn't have ended that better myself. So thank you for that, Parrish, very much.